so that we can move on to transcription. I just want to spend like 2 minutes, no, 2 minutes, 5 minutes on deep sequencing, right? So deep sequencing, I'm sure you guys have heard that term, right? Thrown out at you. Oh, we can sequence the whole genome. Well, the only way you can sequence the whole genome is in a quick, rapid fire kind of way is this stuff called deep sequencing or pyrosequencing, which sounds like you just set it on fire and hopefully the sequence just magically appears. And that kind of is a little bit like what it is. And I'm not, so the next several slides in your thing kind of walk you through this whole technical challenge that is deep sequencing. We are not going to go through that because you guys, you're going to be doctors. You're not going to be deep sequencers. This is not something that we have to like completely understand to know, oh, what's a deep sequence, right? But what I want to talk about a little bit, just for a few minutes, is what it really is and what it entails, okay? Because it's a really pretty straightforward technique that gives us this enormous amount of information. Okay, so this is actually isolating the DNA from a given individual or a given bug, right? So we do this now to sequence genomes of bacteria and viruses and whatever your favorite bug of interest is. You extract the DNA, so this is DNA sequencing that we're doing. You isolate it, and you essentially fragment it, right? So remember we talked about, look at this, bam, I brought my laser pointer. We talked about using shotgun sequencing, right, to walk ourselves down into little pieces of DNA that could actually be sequenced. Well, this is minus the shotgun, right, minus the walking it down part. This is take the whole DNA, fragment it into bite-sized pieces, reasonable-sized pieces, and then utilize that to do essentially PCR sequencing on that entire genome. So we're actually going to kind of sequence the whole thing simultaneously, which sounds kind of crazy, but you're actually, I'm just going to get to the little thing because it actually makes a little bit more sense conceptually when you're looking at it. So we isolate the DNA and fragment it, and then utilizing these little adapters, we're going to put those adapters onto the ends of each of these sequences. We know exactly what the sequence of those little adapters is. And then we're going to take all of this DNA and hybridize it onto this array, this kind of substrate that just has the complement to those adapters. Okay? So that everything that has adapters on it is going to stick to this surface. Okay, and you're going to dilute it out so that it's not all clumped all together, right? So we're looking essentially for each little fragment of DNA to hybridize kind of away from the other pieces, but to a little adapter sequence. And then using straight ahead, head-on PCR amplification, leading strand, right? You're going to actually amplify each of these using this little bridge technology. Again, we're not going to go through how that actually works. But in essence, what you're going to do is for each of those little pieces that you've hybridized onto this array of just your adapters, is you're going to get these little clusters forming because of this multiple rounds of PCR amplification that you're doing for each of those pieces of DNA. So in other words, each of these clusters is going to represent an amplification of one single piece of DNA that you've hybridized on. Now again, we're not going to walk through how exactly that works and the whole process, but in essence, you're now amplifying each of those sequences by itself, yet all together. right? So in one gigantic reaction on this array, you're amplifying the whole genome, which in and of itself is like mind-blowing that you're doing this for an entire genome, right? Billions of base pairs of sequence you're doing on one array, which is pretty phenomenal, right? Kind of like, whoa, what is that about? Now you can do the entire genome, or you can sequence what we call the exome, which we're going to talk about a little bit more soon, right? Or the regions of the genome that are just protein coding, right? That obviously is going to be a large reduction in the amount of DNA that you're going to be sequencing. But that obviously is to just get at, are there particular point mutations that are within the coding sequence of the genome that might be causing disease X or disease Y? So it's a more streamlined sequencing approach to just be looking at the exons and what is encoded there. Okay, so, and then the kind of, to me, the coolest, second coolest part of this is that you actually can sequence these again in situ you can get a sequence of each of these little clusters using what's called pyrosequencing. So you're throwing in a gamish of nucleotides. That's on the next one, I think. You're throwing in A, C, G, and T, right? But these guys have a modification, again, they're dideoxys in essence, so that you're just throwing one nucleotide at this, and it's hybridizing, and because it's a different color, you can actually run it through a scan and say, okay, for that cluster, this is the nucleotide that comes first. For this cluster over here, might be a totally different nucleotide. For this cluster, totally different nucleotide. And then, these are cool because they're kind of swing modifying nucleotides, so you hybridize them, and then you can't add anything else. And then you do this little presto changeo light hitting, and it switches the nucleotide so you can add just one more onto it. It's really insane, but it's cool because in essence you're reading that sequence one nucleotide at a time for each of these little clusters of DNA that you've got. Then you gotta, right, do the whole crazy, oh, look, here's all of my sequences. Let's try and put them in the right order, right? And that's, again, utilizing overlapping sequences, right? You do this with multiple different restriction enzymes so that you've got what sequences are overlapping. You can piece them all together. You throw that at the bioinformatics guys and say, here, get me my sequence. And you can do this whole sequencing. And again, this doesn't take that much. You can get some of these. That's pretty phenomenally cool. And now, here's what the kind of things, I just want to go back here to get us into thinking about, okay, what's come out of this, right? This is a new technology. Last couple of years, we're really starting to do a lot of this. And here's the thing to me, which is phenomenal. Every single person's genome that you look at has between 1 and 10,000 base substitutions from the template sequence that we sequenced in the big genome sequencing project, right? So 1 to 10,000 differences between my genome and what's his name? Jim Watson's, okay? I don't care, because Jim Watson, he's an okay guy. He's kind of a jerk about some things, but I've actually met him. He's an okay guy. And so, 10,000 base pairs different than him, I don't really, that's okay for me. Right, obviously we talked about this deep sequencing of tumors, right? And thinking about and looking at mutations that might be occurring in particularly protein coding sequences that are only occurring in breast cancer cells, are only occurring in liver cancers, are only occurring in lung cancers, right? Can we narrow that mutation rate down and say, if you've got a mutation in this particular gene, it's going to potentially drive the formation of a breast cancer or a liver cancer or a lung cancer, right? That's incredibly important information information for us to be gathering. And the other part that's cool is that we're starting to look at the mutation spectra of particular proteins in disease states. And because we can see, okay, I've got these particular mutations that are occurring in this given gene product that's going to lead to this disease, now I can engineer that protein to test in vitro, right, express it with each of those different mutations, and start targeting and looking at how do drugs act with that protein. Can I fold it differently? Can I get it to come back online and work properly in a tissue culture system, right? That's a lot cheaper than throwing them into rats or throwing them into other kinds of animals. You can do it in vitro to start testing and modifying your drug design, knowing the sequences that are changing in particular disease states. So it's really powerful information that we're gaining on this. The one thing that I came across again, just in my reading in the last couple of weeks, is thinking about circulating tumor cells, right? So if you think about this, and particularly with this hits home with breast cancer patients, is that the metastasis is in the bloodstream, right? Oftentimes, those metastasizing tumor cells are moving through the bloodstream to get to a different organ. If you can actually isolate those tumor cells from the blood, extract their DNA, and look at and see, are there mutations that are evolving in that person over time? And are there mutations that are evolving to perhaps not allow them to be treated with X drug anymore? They've evolved so that they're resistant to that drug. Well, can we now take that mutation spectra and design some new drugs, again, utilizing those cells in vitro to get personalized drug discovery for that individual, right? This is absolutely the wave of genomics in the next 10 years, is personalized drug discovery, right? How can we treat that patient now? That's really, really powerful stuff that you guys are absolutely going to be in on. Cool for you. Yay. Okay. End of story, unless we have any questions about things, other things in this topic that we want to clarify before we move on. Closing the picture. Okay, good. Yay. Everyone is fully aware and fully understand. Or fully Friday. That might be it too. It's all right. It's all good. You should see the campus over at UI. It's insane. Billions of students roaming about. I could barely get to my office. There is a lot of free music going on, though. We should go over. Yes, I have a question a lot. Sanger sequence. It utilizes Sanger sequence. Yep. So Sanger sequencing is just the original dideoxynucleotide sequence. So big di is just that it's got these 4 different dyes, and you can do it all in one tube, as opposed to doing it in 4 tubes. That was the big advance. Okay. Yay. We're on to topic number 4, which is transcription, right? So one of our basic dogmas, basic principles of molecular biology is that we're taking this DNA that we've got, and in our protein coding sequences, and also in our not protein coding sequences, it turns out, we can make an RNA template from that double-stranded DNA, right? We have the capability, our RNA polymerases can read double-stranded DNA or single-stranded DNA and decode that into an RNA string, right? That's basic principle of transcription. Okay, so we're going to talk about the different types of RNA and those to get a little bit into Pol 2. So RNA polymerase 2 in our cells does the transcription for mRNAs, right? For messenger RNAs that are going to be encoding proteins. We have to talk about how that is regulated, right? Because we'll see next week as we get into gene control and the regulation of all of this process that it is exquisitely controlled in our cells. Transcription of RNA Pol 2 has a lot of players involved and a lot of regulatory mechanisms that we're going to talk about. We're going to talk real specifically about the proteins that are binding to what we call our enhancer sequences that are going to either increase, jack up transcription, or downregulate transcription, depending upon who they are, where they're binding, what we want to do. And then we want to absolutely talk about all the modifications that are going to occur to those RNA, mRNA transcripts in particular. Capping, polyadenylation, splicing, editing, turns out. We're going to throw that in at the end, because that's a really cool process too. This stuff is only occurring to our mRNAs, right? So ribosomal RNAs, tRNAs, they don't get all this modification. Right, here's the forest. mRNAs, highly controlled. There's lots of players. The levels can absolutely vary from cell to cell and across development. That's really important for us to keep in mind. And then that they are highly processed. Okay, so let's talk about our different types of RNA that we've got here. Sorry, I'm converting to the little red bouncing dot. Is anybody red, green, colorblind? That you know of? Okay. Can you see this? Can you all see the red dot? Okay, then nobody's red, green, colorblind. Right, so first off, right, and actually the least abundant of the RNAs, but arguably, some would argue this, most important, are the protein coding mRNAs. But we've got, obviously, ribosomal RNAs that are the basic structure of the ribosome and are catalytic in nature. tRNAs, which are the guys that are going to transfer the information or decode the information in the mRNAs into proteins. Small nuclear RNAs, small nucleolar RNAs, right? So I was reading this big thing about nucleoli. Nucleoli, they're the wave of the future. There's all this control going on inside nucleoli, turns out. You'll hear more about that, I guess, somewhere in the next 5 years. And RNAs, right? So we talked and touched about this, and we'll talk about them more as we go along here. The microRNAs and the long non-coding RNAs that are encoded now, potentially in non-protein coding sequence, but in sequence throughout the genome, and that are going to have absolutely tight regulatory control on our mRNA expression, right? So long non-coding RNAs and microRNAs are regulating whether or not those mRNAs that we just finished making are going to actually be expressed, right? So this is a very new and expanding field in terms of studying these microRNAs that microorganisms produce, right? So viruses produce them, bacteria produce them, protozoa produce them, that actually control the landscape of the cells that they're going into, right? So I don't like that protein being around. It's going to inhibit my replication. Turn on that microRNA, shut off that protein, bing, bang, boom, you're done. You're replicating like crazy. My virus does that all the time. It's actually kind of annoying right? To figure it out. Okay, let's think about this, right? In our concept of plus strand, non-strand, average strand, coding strand, purple strand, sense strand, plus strand, alt strands, right? We got lots of different terms for the same exact thing. So let's think about it for a minute. Okay? So RNA polymerase general, is reading just one strand of the double-stranded DNA. Right now, some genes are encoded on one strand. Some genes can be encoded on the opposite strand. They can be going in different directions. Completely depends upon the protein of interest. However, there's always one strand that's going to serve as a template and one strand that is the opposite of that, the complement of that, right? So the template strand, think about this, right? This is the strand that's going to be copied. That is the opposite of the mRNA. So what do we know about the mRNA? What is the polarity of the mRNA? Plus. So the template is minus, right? So the template is minus. It's the opposite of the mRNA. So what does that make the coding strand? It's plus, and it's the same as the mRNA, right? It's the same sequence as the mRNA, except for it's got U's instead of T's. Make sense, everybody? That's something that we need to absolutely keep locked away in our brain. Template strand is opposite polarity, opposite sense, opposite direction. Everything is opposite from the mRNA. Okay? We transcribe, we read that template strand, and we get our RNA transcript, which is the same sense, same sequence, same direction as the coding strand. Just an FYI, when you see, if you've got and you're reading a paper and it says, and the DNA sequence is bloody, 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 and it only gives you one strand, right? It's only giving you one string of nucleotides. That's the coding strand. They always tell you the coding strand, the strand that's going to encode the that's going to have the same sequence as the mRNA that's made in the protein. Just the FYI. Right. So every protein coding gene and every RNA encoding gene is going to have the basic structure of this, the transcriptional unit, we would call this. Okay, so here's the gene of interest that we're wanting to transcribe. We're wanting to make an RNA from. There's always a place where the polymerase, be it RNA call 1, 2, or 3, sits down and starts transcribing, starts making a string of RNA. That's called the start site. It's never going to vary. That is what it's called, transcription start site. In front of that, and these are on eukaryotic promoters now, is what we call a promoter sequence. That's where the machinery that's going to help the polymerase sit down recognizes this piece of DNA as something we want to transcribe. Okay, so you can think about it, right? The promoter, he's the guy that talks everything up, right? That makes it big, that says, oh yeah, this is where you want to come, right? This is what the promoter sequence is. Come over here, big old flag, sit down over here. That's what the promoter sequence does. Now, in the case of Pol II transcripts, of mRNA encoding transcripts, like we said, we're going to have proteins, what we call specific transcription factors, that are going to enhance or limit the basic expression off of that transcript. Okay? Enhancer regions are where those transcription factors sit down on the DNA. Okay? These are oftentimes what palindromic sequences, again, they don't have to be palindromes, it can be, but they oftentimes are. And the thing about enhancers that you need to remember is they are distance independent, okay? They can be 10 kb away, 20 kb away, and we'll see why. It's because the DNA can be really flexible, and you can actually move these proteins around to be close to the promoter sequence by just bending the DNA around. So they don't have to be right next to where the polymerase is going to be sitting in order to have an effect. So they're distance independent, they're place independent, and they're also direction independent. Okay, so it can be that the enhancer is like this, and the protein sitting down like this. It can be completely flipped around, and the protein is still going to recognize it and sit right down there. So enhancer sequences are sequence direction independent. They'll sit on that site regardless of whether it's flip-flop. Somebody had a question.
Yeah, so that being the case, can they do the enhancer for multiple promoter regions, or is it typically just one?
Each promoter generally has its own enhancer sequence. Now, there are exceptions to that rule, but yeah. Because we're really trying to regulate that particular gene product at this point. Now, that being said, the same type of transcription factor is going to have these enhancer binding sites all over the genome. Okay, so many, many, many proteins can be regulated by the same transcription factor. Make sense? Okay. Nicholas, did you have a question? Oh, sorry. Okay. Yes? Thumbs up? Okay. Right. Now, I just want to utilize this as a thought of what has to actually go on in terms of the DNA here. Because what's pictured here is actually what goes on in the prokaryotic system. Okay? In that, yes, absolutely, the polymerase is going to sit down and come into contact with the promoter region. That region is going to be opened up, right? It's going to be melted, is what we call it, that promoter sequence. And the polymerase is going to start initiating polymerization. But as it's pictured here, this polymerase is actually binding directly to the DNA all by itself. Presto, changeo. Not happening in our system, okay? There are like 10,000 proteins that help the polymerase settle down onto the transcript start site, okay? So bacterial polymerases have sequence-specific recognition and the capability to actually melt the DNA and open up the promoter sequence. We can target that. Yay, us versus them, right? Because there's a drug called rifampicin, or a bacterial antibiotic called rifampicin, and it's in your notes already, that targets the polymerase of the RNA polymerase of bacteria and targets this specific selectivity of being able to bind and see where the promoter is. So that obviously, right, is not a component of our polymerase, so it's not going to target us. We don't have that capability. Okay, so that's a cool us versus them thing as well. So we're going to bind, and we're going to have help. We're going to melt this sequence. Again, we're going to have help doing that. And we're going to initiate transcription at the transcription start site. The polymerase, once it's polymerizing, doesn't really need much help. It's got its own deal there. It's going to chug along. It does need what in front of it? A helicase. Right. Remember, we're still doing that same thing. We're opening up the two strands. Got to have a helicase in front of it as well. We're going to polymerize here, and then we're going to come to the transcription stop site, which we'll talk about in a few minutes. How that's recognized. The polymerase falls off. The transcript gets cleaved out of the polymerase and is released. All the modifications are going to have already happened, as we'll see. This is a co-transcriptional modification process. Gets shipped off to the cytoplasm to hopefully get made, or maybe to be silenced by the microRNAs that are hanging around at the nuclear core waiting to grab it. Okay, so basic process of transcription. All right, let's do a little exercise. And we'll just throw these out there. How many polymerases do prokaryotes? This is RNA polymerases. How many RNA polymerases do prokaryotes have? One. Yes. One. Put a one there. How about for us? Three. Three. RNA 1, Pol 1, Pol 2, Pol 3. everybody does different things. Paul 2 is the guy that we really want to remember because he's the one that does the mRNAs and the microRNAs and the long non-coding RNAs. Paul 1 and Paul 3 are involved in ribosomal RNA transcription and tRNA transcription primarily. Recognition of the promoter sequence, what we just said. The RNA polymerase of the prokaryotic system has its own capability of recognizing all by itself. So check, yes, self, whatever you want to put in there. Eukaryotes, no way, man. It's got this big, gigantic complex of proteins that's going to help it to recognize the promoter sequence. How about coupling of genes, right? So what I mean by that are there these things called, what? Operons. Thanks, Aaron. Thanks for giving me that hint. Operons, right? So, in other words, is there a string of genes that we know is involved in all kinds of things, but one particular process? And do we string all those genes together and just have one promoter that makes all those genes all at the same time? No, we absolutely don't. Everybody gets their own promoter, right? Everybody gets their own cell phone. Everybody gets a blue ribbon. Everybody gets their own promoter. Not the case, absolutely, in prokaryotes, right? The operon units are very, very coupled and strongly coupled so that, right, they can say, I need to metabolize this sugar right now, and I need these 6 genes, these 6 proteins to do that. All right, I'm just going to turn that all on. I'm going to metabolize them all. I'm going to have the same exact amount of each one of them. Now I can metabolize that sugar and go along on my merry way, right? We don't want to do that. We don't want to do that. We want every single protein sequence to have its own promoter so that we can turn up or down the thermostats appropriately. Okay? Coupling of transcription and translation, right? So in other words, as soon as I make an mRNA, is it translated? Me. No, absolutely not. How about in a bug? Bacteria. Yeah, right? And why is that? Yes, we have a nucleus. The RNA has to get transported out of it. It also has all kinds of modifications that we are going to talk about. Our transcripts are modified ridiculously heavily, not so much in the prokaryotes. In fact, not at all. Okay? And that's because as soon as it comes out of the polymerase, it gets sucked into a ribosome, right? So in other words, the ribosome is just sitting there waiting for that transcript to come out of the polymerase. It's so hungry, it doesn't even wait till it comes out the entire thing, right? As soon as it comes out of the polymerase, even a tiny little bit, it grabs on and starts translating. It's ridiculously fast. And then, as soon as the ribosome is done, the damn thing gets eaten. So the mRNAs are ridiculously unstable in a bacteria. In our cells, they're very, very, very stable. Depends on the transcript of interest. But they're much, much stable on the order of hours, sometimes days, that a single mRNA transcript can be around. That all has to do with that poly A tail that's stuck on the end of it and how it's not able to be degraded over time. Okay, so we have a lot more processing and information that goes on to our RNAs, particularly our protein polymerase. So this is just a little schematic that I want to point out a couple of things on. Okay, so we talked about how Paul 1 and Paul 2 and Paul 3, RNA, Paul 1 and Paul 2 and Paul 3 are all working on different transcripts, right? And get to that for a second. So Paul 3 is mostly mostly working on tRNAs. Pol 1 is mostly working on ribosomal RNAs. And then Pol 2 is our mRNAs and our microRNAs. Okay? So, but as we just said, regardless of the polymerase, there are proteins that are going to help it to recognize where it needs to sit down on the DNA. Okay? So again, the polymerase in and of itself does not have recognition capability or binding directly to the DNA capability. Okay? So how then is it going to actually find out where it needs to be? Well, there are what's called transcription factors, TFs, transcription factors, that are going to help the polymerase to see where the DNA is that it needs to sit down. Each polymerase has a different set of factors. Again, we're not going to go over who's who and what's what and all of that good stuff, because you don't need to know all those names. However, you can see that each polymerase, right, has its own set of factors that it goes along with it. So Pol 3 has the transcription factor 3 binding group. Pol 2 has the TF2 binding groups. And Pol 1 has the 1 groups, right? So this is SL1. It's one of the major components that is utilized for Pol 1. Okay? So each of the polymerases has a grouping, a complex of proteins that's going to help recognize the promoter. That's TF3B, SL1, or TF2D, depending on which polymerase. But I want you to notice that each of these complexes, these guys that are helping to recognize the promoter, have this protein called TBP, the Tata binding protein. It is an essential protein for all these polymerases to recognize the promoter site. Okay? So, as the name implies, the Tata binding protein, it binds to the Tata box, that region of the promoter that's only present in RNA Pol 2 transcripts. Okay, our mRNA encoding transcripts. That's where it was first found. So that's why its name is Tata binding protein. It binds directly to the DNA in Pol 2 transcription. Okay, it binds to that Tata box. In Pol 1 and Pol 3 transcription, the TBP protein is not the protein that binds to the DNA and recognizes the promoter sequence. That is other components that are in these complexes. However, TBP is there to act as the interface with the polymerase. Okay? So TBP is the guy that's always in direct contact with the polymerase itself. Okay? So that's why it's so absolutely important for transcription, because it's the one that brings the polymerase into register with the DNA. Okay? Whether by the TBP binding directly to the DNA or another component of the complex binding directly to the DNA. But the TBP is the interface to the polymerase. Okay? Regardless of the polymerase we're talking about. Right? So that's why this is here. Just to recognize that, and recognize that I want to throw this term out at you because it's important. These complexes, all of these complexes of proteins that are trying to help the polymerase to bind to the promoter are what we call basal transcription factors, okay? Or general transcription factors. They're the same thing. But basal transcription factors are the guys that are absolutely necessary for transcription to occur. They have to all be there, or we are not going to get this polymerase settled down on the promoter. Okay? So the basal transcription factors are absolutely required. They're the guys that bind to the promoter. The enhancer binding transcription factors are the auxiliary ones, if you will. The specific ones that will not always be bound. That's going to be the ones that vary according to the transcription. Does that make sense to everybody? So basal is always there. The specific guys, the guys that bind to the enhancers, not so much. They're going to vary. angle of looking at call 2 transcription, and really we've already covered this in terms of the TATA box in that promoter region is going to be bound by TBP, and the rest of that complex is going to help the polymerase bind and start transcribing, right? And these enhancer sequences can be upstream, they can be downstream, they can be within the transcript itself, doesn't really matter. So there's a term that I want you to think about and to keep in your head, upstream and downstream. Those are things that we're going to utilize a lot for the next 2 weeks. Right, so upstream is the sequence that is 5' if you will, to the transcription start site, right? Because remember, again, polymerization is always going to be 5' to 3'. So to the left, upstream is 5' to the transcription start site. Oftentimes enhancers are there, but they can be what we call 3' to the transcription start site. They can be downstream of the promoter sequence, or they can be inside the coding sequence itself. Exons and introns, we'll talk much more about that in a little bit. Okay? So I want to walk through for a minute how the polymerization of call to transcripts starts. Because, again, it's a ridiculously complex dance. I don't expect you, again, to remember all these players, but I want you to take away from this that there are hundreds of proteins involved in this. So each of these little globules that we're seeing here that are representing transcription factors, basal transcription factors, are actually a complex of proteins. So each of these transcription factors are groups of proteins together. So this is an enormous complex of proteins that is just designed to settle the polymerase down on the transcript, right? So this is a big dance. This is a big production to get this guy settled down. They occur in a sequence of events. And again, you don't need to remember who comes first, who comes wherever. Except that TF2D comes first, right? Because that's the guy who actually encodes TBP, our TATA binding protein. So the TATA binding protein has to come in first to find the TATA box. If it's not there, then we don't know where to start our transcript, right? So the TBP has to come in, recognize the TATA box, and settle down. So that now the polymerase can be put into register right in that sequence. Okay? So TBP comes in first, then several other complexes, again, which you don't need to really know who's doing what here. There is another complex of proteins called TF2F, which in essence brings the polymerase into register. So I like to think of TF2F as like a tugboat. Okay? So think about when you're in a port, right, and you've got these big gigantic boats, ocean liners that are out floating around in the ocean, and they need to come into port. But they don't know how to get to the dock that they need to get to, right? And they can't motor around, right? These things are way, way too big to maneuver all on their own. So the tugboat actually goes outside the harbor, meets this big gigantic boat, and ferries it in, directs it into where it needs to go, into the port. So TF2F is in essence the tugboat as bringing the polymerase into where it needs to be. To port. It's bringing it home. I'm the king of mountain. TF2F, tugboat. Now, Paul 2 can directly contact TBP. He's here. Now, TBP is bound to the DNA. Paul 2 binds to TBP. It's in register. It knows where it needs to start transcription. There are some other complexes that need to come into play here. We're just back up on top here with the polymerase. TBP is here. TF2F, our tugboat came in. Now I want to focus on TF2H for a second. This guy right here, this orange one. TF2H is the other component complex that you need to remember. So TF2D has TBP in it. TF2H has a multitude of proteins in it that are insanely important for polymerization to start. Okay, polymerization to start. Okay, because it has three distinct things that it does. So TBP is a kinase. Okay, so what do we do? What are kinases doing again? Adding phosphates, right? This time we're adding phosphates to a protein. It has helicases, bless you, that are going to help unwind the DNA. And it has an ATP, okay, that's going to kick the polymerase into gear and help start it in the polymerization process and help to open up the strands. Okay, so it's going to fire this promoter. How does it do that? It does that primarily, well, probably the most important thing is that in its kinase capability, it phosphorylates directly the polymerase. Okay? So RNA Pol 2 is phosphorylated by TF2H, by the kinase in TF2H. Why is this important? And just to clarify this, it phosphorylates the C terminus of the protein, right? So the very, very tail end of the polymerase. This process is insanely important because the phosphorylation is going to allow this polymerase to be recognized by all of the post-transcriptional modification machinery. The splicing machinery, the capping machinery, the polyadenylation machinery is all going to recognize this phosphorylated form of the protein. If it is not phosphorylated, it will not transcribe and it will not be allowed to be recognized by all this post-transcriptional modification machinery. Okay, so phosphorylation of this protein is absolutely essential for it to function properly. Right? So that means TF2H is insanely important in this mega complex that we're assembling here on the promoters. Okay, so its helicase activity is going to help start to unwind the strands. It's going to chug along with it and help to continue to unwind the DNA. And the ATPase, again, is going to help to provide the energy for this polymerization and this whole big production, if you will, to continue along. Okay? 2 o'clock. Let's take 10 minutes. And we'll continue on with our saga of transcription, talking about some of those modifications that are going to occur.